Welcome to the 39th Annual Florida State University Young Scholars Program Research Symposium. Before we get started, I'd like to introduce myself and thank a few of the people who make the program possible. I'm Erica Staling, the Associate Director of the Office of STEM Teaching Activities and Co-Director of the Young Scholars Program. And with us today, we also have Barbara Shoplock and Hannah Heister, who are also co-directors of the Young Scholars Program, and whom I would like to thank for working tirelessly to make this year a success. We all truly wish we could have been back together on campus this year, but everyone involved did a phenomenal job making the most of a difficult situation and making it work remotely. As most of you likely know, typically young scholars live on campus and engage in program-specific coursework, research, and enrichment activities in person over the course of 6 weeks. This year, students participated from their own homes, and we offered 3 mornings per week of program-specific academic courses and 2 mornings per week of open access research seminar through a combination of Canvas and Zoom. Through the Open Access Research Seminar, students worked in peer groups to design and investigate original research questions using primarily open access datasets with a variety of structural supports. The students learned how to access and work with these datasets through a series of seminars from guest speakers and received mentorship from FSU faculty, postdocs, and graduate students. Today's symposium is a culmination of that work. Although we are focusing on open access research this afternoon, I'd be remiss not to also thank our academic course instructors, including Drs. Garai Octon and Alexander Resnikoff from the Department of Mathematics and Kyle Shaw from the Department of Scientific Computing. I'd also like to thank the Director of the Office of STEM Teaching Activities, Dr. Ellen Granger, the Dean of the College of Arts and Sciences, Dr. Sam Huckabee, and FSU's Provost and Executive Vice President for Academic Affairs, Dr. Sally McRory. Thank you to all of the FSU faculty and administrators who continually support the Young Scholars Program and enable us to provide this unique opportunity to the best and brightest high school students from all over the state of Florida each year. We have 8 teams of students who will present today. Each group will have 10 minutes to present their work, and then there will be roughly 2 minutes for questions. Dr. Heister will introduce the groups, and then I will facilitate the questions by monitoring the chat. If you have a question, please feel free to either type it directly into the chat and I can read it for you as time permits, or you can raise your hand or type, I have a question, and then I may call on you from there. So without further ado, I will turn it over to Dr. Heister to introduce our first presentation while the students get their slides up.
Thank you, Dr. Staling. I'm also going to share in the chat a link to the program. So if you don't have a copy or would like to look there, you can click on that and get a PDF of the program. Thank you. So like Dr. Staling, I'm really excited to hear from our students and to see the culmination of all the hard work that they've been doing. Our first group worked with data from the National Ecological Observatory Network and received mentorship and support from Professor Tom Miller's lab. Especially from Danny Davis.
All right. Hello, everyone. My name is Jin Kwan. My group members, Talia Givoni, Andrew Busby, Odin Farkas, Deepika Kanan, and I studied the effect of nitrate concentrations on macroalgae populations. So to put our research project and its applications into perspective, it's important that we first understand the nitrogen cycle as well as eutrophication. So figure 1 shows us the nitrogen cycle, or the flow of nitrogen through nature, in which nitrogen travels through and is converted across many forms, ranging from nitrogen gas to nitrates. When nitrogen flows through the cycle naturally, the ecosystem maintains its balance. However, nitrates are also often used in fertilizers, and extra nitrate runoff amongst other chemicals, often from farmland and other agricultural sites, can cause an imbalance and lead to many complications. And that brings us to figure 2, which shows us eutrophication. Eutrophication is one of the biggest complications of this excess nitrate runoff. It is the phenomenon that occurs when excess nutrients cause an oversaturation of plant and vegetation growth in bodies of water. This excess plant life can block sunlight and cause depleted levels of dissolved oxygen, or hypoxia, which can make the aquatic environment essentially inhabitable for fish and other organisms. We decided for our project to focus specifically on nitrates in relation to macroalgae growth, all in the context of proximity to farmland. We used 4 bodies of water as test sites, 2 closer to agricultural lands and 2 further away. We decided to conduct research on this topic in order to analyze the relationship between these 2 factors in the context of human impact, as showing a link would help us point out and mitigate this human-induced destruction of our aquatic ecosystems. And now I'll pass it off to Andrew to talk about the significance of our past literature.
Hi. So this first study by Driscoll et al. Concluded that in New York, acidic deposition, that is the acidification of soil and surface water, is linked to a decrease in diversity of aquatic organisms, an increased accumulation of mercury in fish, increased eutrophication of coastal waters, and enhanced stress to certain arboreal species. This is relevant to our study because it shows how water quality can have a significant impact on local ecosystems. This second study analyzed the correlation between the growth rate of a population and the water quality of a river. The researchers utilized the Bayesian network classification model to estimate the population capacity based on a known water quality in a region. As we discussed with eutrophication, this capacity has a measurable impact on the animal populations living within those bodies of water. The third article describes the National Ecological Observatory Network datasets in detail. The authors elaborate on several topics, including, but not limited to, the depth of the data on the NEON platform, the methods used to collect the data, and how the data may be used for further scientific research and to answer questions. This was useful to us in understanding these NEON platforms that we would be using for our research, namely in their applications and limitations. I'll hand it off to Talia now to describe our research question.
So analyzing the previous literature, as Andrew has just described, prompted us to ask the following scientific question. How do various nitrate concentrations affect macroalgae populations? And moving on to our hypothesis. If nitrate concentrations increase, then macroalgae populations will also increase. We predict this because previous research has shown that nitrates can boost macroalgae growth, so predicting a positive correlation with agricultural lands. And now I'll describe our field sites. So for our research, we chose to analyze 4 bodies of water, 2 that are closer to agricultural lands and 2 that are farther away. On the left-hand side, we have a creek and a river that's closer to agriculture, and on the right, we have the same comparatively, but those that are farther away. So figure 4 in the top left shows our first field site, Kings Creek. Kings Creek is located in Kansas within the Prairie Peninsula range. As a tributary of the Kansas River, Kings Creek has faced periods of drought, unfortunately, and erosions, and is home to 2 underlying stream terraces and alluvium deposits. So shifting down to the bottom left corner, we have the Aracari River in the Central Plains region of Colorado. The Aracari River is actually super long, over 156 miles long, and actually spans across 3 states. Yet we are focusing on the section in Colorado that the NEON data site has. It is actually home to many Great Plains animals, such as birds and amphibians. Now moving to the top right corner, we have figure 6, which is Tennessee's the Conte Creek. The Conte Creek is part of the Appalachians and the Cumberland Plateau region. This stream has varying depths, ranging with boulders to lush vegetation, and the aquatic area is in the northwest part of the Great Smoky Mountains. Our final figure 7 in the bottom right depicts the Blue River of Southern Plains, Oklahoma. The Blue River spans wide and is actually most famous for their fish, specifically different breeds of trout and catfish. And so I will now pass it off to Deepika to discuss the methods by which we went about conducting research on these field sites. Thank you.
Yeah, so to start off with our methods, the first thing we did was determine which field sites were in common between both the nitrates and the macroalgae data. And once we determined which field sites were in common, we took screenshots of all the different field sites with a radius of 60 miles. And you can see some examples of the figures that we used here. And after that, we had to determine which field sites to use by calculating the percentage of agricultural land around the fixed radius from each test site. And the agricultural land was shown by this certain shade of brown, which we can see here. And we wrote a Python program that determines the percentage of that brown on the given land. And this allowed us to quantify the actual amount of farmland. So as we can see in both figures 8 and 10, we had a greater percentage, but we had a smaller percentage in figures 9 and 11. So we decided to use those 4 so we had more diversity in the amount of farmland. So after that, we wrote a Python script to download the NEON data, and we downloaded it for both the nitrates and the macroalgae. And then we also recorded the data for the macroalgae count for different times on the same day, because NEON had like different times on the same day they would record. So we calculated the average per day. And we also calculated the average nitrates for 30 days prior to that macroalgae count. And we did this because macroalgae takes 1 month to grow. And then we graphed the macroalgae count versus the nitrate count for each field site, so we can see this relationship. And then we also graphed the macroalgae and nitrate counts for each field site to the farmland percentage. And this allowed us to see the relationship between the agricultural land use and the macroalgae. So now I'm going to pass it off to Odin to discuss our results.
So to look at those relationships that Deepika was mentioning, we plotted nitrates versus macroalgae populations first. As you can see on all four graphs, our x-axis is the prior month concentration of nitrates on average in micromoles per liter. And on the y-axis, we have the macroalgae counts on the day of measurement. On the left, we have our sites that had low agricultural density, and on the right, the ones with high agricultural density. On the top, we have our creeks, and the bottom, our rivers. Despite all those differences, something you will notice is that all have a weak positive correlation and similar slopes on their trend lines. Also, all four graphs shared a similar quite low R-squared coefficient, something we'll touch on later. On the next slide, we next reviewed our initial ideas about agricultural density and its contribution to nitrates and macroalgae in the water. As you remember from what Deepika mentioned about our sites and the agricultural testing there, we predicted that sites with higher agricultural density would have higher concentrations of nitrates. So in figure 16, you can see that we plotted land cover versus the average nitrates over the time at the site. And we actually found the reverse correlation of what we expected with a pretty strong R-squared coefficient and a negative slope on that line. What we initially explained this is, how we initially explained this was we believed that there might be lower average nitrates in the areas with higher density because macroalgae consumes these nitrates. So what we did expect was in the next graph, we would see more macroalgae in these regions. However, upon plotting land cover versus average macroalgae, we didn't see that result. And we actually saw the same negative correlation, leading us to believe that in the context of our study, the areas with higher agricultural density actually had lower levels of nitrates and macroalgae, something we did not expect based on our background research. On the next slide, you'll see because the agricultural sites and sites with lesser density were clustered into similar areas of coverage. We clustered them into agricultural and natural sites and plotted them on a bar graph for ease of viewing, but it's the same data we just described on the last slide. Next, we can go into some of the limitations of our study. The main thing is that NEON data is somewhat inconsistent in the way they report between different data sets. Because we had to use nitrates and macroalgae data sets, although some are collected at the same sites, they're not always collected on the same dates. For example, we may have a day where macroalgae data was collected, but for the months prior, there was no nitrate data. So we were unable to create that as a point on our plots, thus leading to a somewhat low R-squared coefficient because our data could not compensate for some outliers. Also, we were unable to fully understand confounding variables with just the topographical data we were provided by NEON. As you can see, we did see how agricultural density was in the region, but there are many other things that contribute to nitrates in water, such as a wastewater treatment plant on the edge of a river that could easily contribute to different nitrate levels than we would have expected. So, despite a positive correlation on some of our graphs, due to a low R-squared coefficient, our results are not statistically significant enough to determine that our hypothesis was supported. However, in the future, we hope to conduct future research on both the environments of the sites we tested to get a better understanding of what other features may influence nitrate concentrations in these streams, and also test our initial hypothesis about areas with higher agricultural density leading to higher concentrations of nitrates and higher populations of macroalgae in the tens of other NEON field sites that exist. On the next slide, you will see our work cited, and I will pass it off to Jin to wrap us up.
Finally, thank you so much to everyone who helped us throughout our project and this program, but we'd like to extend some special shout outs to Dr. Erica Staling, John Suter, Danny Davis, and our YSP program directors, counselors, and professors. Thank you.
Thank you so much for the interesting talk and for the thank yous. Our audience can use Zoom reactions to give a round of applause while we open the floor to questions. So remember, you can either type your question into the chat and I can read it, or you can raise your hand or chat, I have a question, and just be ready to speak. So I'm seeing lots of applause. Do we have any questions? Okay. Okay, I have a hand raised. Go ahead, Sandhya.
A great presentation. I was just wondering what other factors you think could contribute to microalgae or macroalgae growth within your locations, if not nitrates.
Yeah, I can take this one. So we know that agriculture often has a big impact on nitrate concentrations because of the fertilizer, but it also comes from other places such as pastures and with emissions from cities. So those are some examples of variables that likely could have affected our data, but we didn't necessarily have the ability to take all of that into consideration at once.
I would also like to jump in and say phosphorus as well. Phosphate in the water could also is also a nutrient for macroalgae. So runoff from that could also increase the population of macroalgae.
Thanks so much. We do have time for one more question. Let's see. So we have a question from Kyle Shaw in the chat who asks, did you get your images from NEON or somewhere else for those images of your agricultural density?
I can take this one. So some of the I'm not sure if he's referring to the actual screenshots of the brown versus the green, because that was from the NEON field sites. But if he's talking about the other images throughout the presentation, some of them were from, especially for the field sites, we had specific details that I described about field sites, like different aspects of the environment and the animals. A lot of the images actually came from those sites, and they're in our works cited.
Awesome, thank you so much. That's all the time we have for questions, unfortunately. And so Dr. Heister will introduce the next presentation as those students get their slides up.
Great. Thank you, Dr. Staling, and thank you again, Group 1, for that great presentation. So our second group worked with structural, oh, excuse me, I think my camera slid off for a moment there. Our second group worked with structural biology to study variants of the novel coronavirus, receiving mentorship and support from Professor Beth Stroop's lab, especially from Dr. Stroop and George Maguire. So I'll leave it to you to take it away.
Good morning, my name is Andrea Ortiz, and along with my group consisting of Aryan Gala, Nicole Rashani, Rania Sevillana, and Sonika Prakash, we will be presenting our research in relation to the effects of mutations in the SARS-CoV-2 variants on viral infectivity. To start, we narrowed our topic to investigate and explain which interactions between the different spike proteins of the SARS-CoV-2 variants and the ACE2 human receptor affect the infectivity of the virus. For a little bit of background, the SARS-CoV-2 is a virus that's single and positively stranded, and it's an RNA virus whose genome encodes 4 structural proteins, including the spike protein, the envelope protein, the matrix, and the nucleocapsid. The spike protein is a type 1 fusion protein composed of 2 subunits, with S1 being responsible for receptor binding and S2 for membrane fusion. The SARS-CoV-2 virus uses the ACE2 human receptor for entry into target cells, and therefore the S protein determines the infectivity of the virus and its transmissibility to the host. In our study, we decided to investigate the 4 variants of concern in the United States as classified by the Center of Disease Control and Prevention. And these include the alpha, beta, gamma, and delta variants, which have a series of mutations with the potential of affecting the conformation of the spike protein and that therefore binding and infecting itself with the ACE2 receptor. So it is known that a stronger attraction between the ACE2 human receptor and the variant spike protein will lead to an increase in infectivity. So we predicted that factors like shorter bond length, a greater difference in surface charge, and chemical complementarity of amino acids in this binding site will produce the most infectious variant, and that these would be accurate measures of infectivity.
So before we go into the results, we think that there must be some additional information covered to be effectively introduced. So first of all, we're going to be going over bond distance comparisons, which can allow us to analyze the interactions between spike protein and ACE2 receptor. So the concept is that the shorter the bond distance, the higher the infectivity. So we will introduce this in terms of p-value, meaning the lower the p-value there is, the more statistically significant the data is, which means that there will be higher infectivity in terms of our data. Moreover, we will be doing sequence alignment. So we will be comparing the sequences of various variants and analyzing the mutations and the consequent chemical changes that they bring. So the concept behind that is that there will be different R groups from the new amino acids, and this will bring new properties. So this can affect the orientation of each molecule and thus affect their interactions, conformation, and overall structure of the region, thus affecting the infectivity as well. Moreover, we will be going over surface charge comparisons, which is another form of interaction analysis, and the the reason behind that is that the greatest surface charge difference between the ACE2 receptor and the spike protein, the higher the infectivity there will be. So the methods that we went into was that, first of all, we downloaded the sequences and structures from sources like NCBI and RCSB, completed the sequence alignment and chemical analysis of the mutations, as I mentioned previously. We modeled the variant structures in 4 sessions of Chimera X, which is a software that we will introduce. And this was done through a modeling software of Swiss modeling. So we created our own model through the variants. We matched the ACE2 receptor with each variant structure in each session. We use Chimera X investigation tools for bond distance, surface charge, and more, and then analyze the interactions considering all the aforementioned criteria.
So in our first analysis method, we conducted a multiple sequence alignment comparing the 4 variants of concern to the original SARS-CoV-2 viral spike protein. From this analysis, we make 2 main conclusions. Firstly, mutations do not occur in one section of the spike protein. Rather, they are spread throughout the protein itself. And secondly, when these mutations occur, they occur in clusters of 2 to 3 amino acids in a row, signaling that mutations happen together at a time rather than individually. We use the identified mutations from this sequence alignment for further amino acid analysis.
Continuing on from Sonica's point, we use the mentioned sequence alignment to identify the amino acid mutations in all of the variant spike proteins compared to the spike proteins of the original strain. For an overview, the properties of these amino acids are very crucial to analyze when studying the properties of the protein overall because of the fact that these amino acids influence the structure of the protein and more specifically the folding because of the intermolecular forces between the amino acids. Overall, we can see here through the tables the different amino acid mutations and the changes in properties of those amino acids. Among these 4 variants, there are many amino acid mutations that are exactly the same, including occurring at the same position of the spike protein among the 4 variants, and many similar mutations where a specific amino acid is changed in more than one of the variants. There are also instances such as in the alpha variant where a few amino acids are deleted. So as for the analysis of these amino acid mutations when looking at all 4 of the variants, we can see that these changes, although minor compared to the total amino acids that make up the spike proteins of the variants, are what result in the different properties of the spike protein and thus the different levels of infectivity of the virus. In addition, another interesting thing we found when comparing the different mutations to each other is that a lot of these variants have very similar, if not exactly the same, amino acid mutations. And this may indicate that these amino acid conversions are what create a specific conformational change that make these variants more effective and more infective than the original strain.
So next in our analysis, we used Chimera X to determine the bond lengths of the 8 hydrogen bonds that bind the viral spike protein to the human ACE receptor for each of the variants and also the original virus. We then conducted a 2-sample t-tested means between the average bond lengths of the variants to the original. At the alpha equals 0.05 level, delta and gamma had a statistically significant smaller average bond lengths than the original spike protein. Thus, this analysis ranks delta and then gamma as the most infectious. We then used BioPython, a Python library for protein analysis, to determine the isoelectricity and aromaticity of each variant compared to the original. So firstly, isoelectricity is the pH level at which the net charge of a protein is neutralized. Since delta has an isoelectricity of 8, which is the highest difference than the original and is above the pH level of the normal human body, delta is able to maintain a net surface charge more compared to the other variants. We then reasoned that delta is the most infectious from this because a greater surface charge means greater binding affinity to the human ACE2 receptor. And secondly, aromaticity implies the ability to form additional pi bonds between the spike protein and the human receptor. Beta has the highest aromaticity and thus is most infectious under this characteristic that we analyzed. And lastly, we conducted Coulombic surface charge analysis using the Chimera X software. We determined that the greater the charge difference of the variant from the human ACE2 receptor, the greater the binding affinity and infectivity between the two in our analysis. So using this background and the minimum and maximum surface charges that was identified by the Chimera X software, we determined that the beta and delta variants are the most infectious from this specific characteristic that we analyzed.
So we determined the infectivity of a variant by looking at bond length, surface charge, isoelectricity, aromaticity, and amino acid substitutions. Analyzing these areas indicated that the Delta variant is the most infectious. This is a conclusion that's been agreed upon by several open access journals, which leads us to conclude that these molecular characteristics are correlated with a variant's level of infectivity. The table on the right sums up the results of each analysis. The variants are given a ranking of 1 to 4 in each category, 1 being the result that leads to the highest level of attraction between spike protein and receptor, and 4 being the result leading to the least amount of attraction. Delta took first place in 3 out of 4 areas. The relevance of our research topic can be seen in making vaccines and predicting the infectivity of new variants. We do recognize that our research has some limitations. We used our own list of properties to gauge infectivity of variants, but infectivity can vary person to person depending on their health and medical history. Aromaticity shows conflicting data where beta was ranked first. However, because delta took precedence in 3 out of 4 areas, we still distinguish delta as more infectious. Epidemiological data backs up delta as the most infectious, but when it comes to which variant is next in infectivity and what's next after that and so on, there is some conflicting data. We believe that this is because we studied only 4 molecular properties, and if we were to continue this research, we would analyze more properties. Other ways to extend this research includes finding exactly the significance of each property if one outweighs another in terms of infectivity, and comparing how and if different variants react differently to antibodies. We'd like to thank all of the co-directors and especially thank Dr. Stroop and Mr. McGuire for their time and mentorship.
Thank you so much. So let's go ahead again and use the emojis and the whatever we have at our disposal to give this group a round of applause. It was a very interesting talk, and I'm happy to help moderate questions through the chat or hand raising. Okay, let's see. Looks like we have a question from George McGuire.
Hi, so first off, congratulations on this project, guys, like especially in the time that you had to work on this. Like from what I've experienced, this research and that presentation is at the level of like an honors thesis project for seniors in the biology department at FSU. Like this was great. The amount of information you were able to find was awesome. The way you analyzed it was smart. The way you presented it made it clear. I really enjoyed your presentation. My question for you guys was, you mentioned that many of the variants share multiple mutations between them. What do you think this suggests about the relationship between these variants, like the evolutionary relationship between them?
I could take this question. I think when analyzing the different mutations that they share, a conclusion that we can come to is that maybe the variants evolve off of one another, which is why they share these certain mutations. And as well as we mentioned before, that because they have these same mutations, this can lead us to believe that those specific amino acid mutations is what makes a specific conformational change in the protein shape, and which results in the increased infectivity of these variants.
Awesome. Yeah, I thought that was really cool. Thanks again, you guys. You did great.
Great. Okay, so we have a couple more questions here. Talia, would you like to ask your question?
So based on what you just said in the last question, did you notice a specific amino acid that can cause more mutations or is more prone to mutations?
Yeah, so the one specific mutation that we saw across like 3 of the 4 variants was something called a D614G mutation. So that was a mutation at the 614th position in the protein between these 2 amino acids that was like consistently there against 3 variants. And there have been research studies claiming that like that particular mutation is one that's causing a lot of infectivity. So that might be something to watch out for, like if there are unfortunately like future variants of the spike protein, that might be like consistently holding out through future generations.
Great, thank you so much for the thorough answers. Unfortunately, in the interest of time, we need to just keep on moving. So I'm going to turn it back over to Dr. Heister to introduce the next group.
Great talk, group 2. Thank you again for that fascinating presentation. Our third group worked with epidemiological data to study the spread of coronavirus variants, receiving mentorship and support from Professor Jonathan Dennis. So I will let our third group take it away.
Hello, everybody. Today we will be talking about the trends within the spread of COVID and its variants with Isabella Abad, Lake Tobo, Vishal Suresh, and me, Carl Ryder. COVID-19 has been a highly infectious disease transmitted virally that has led to a global pandemic, and it has disrupted everybody's lives, as you all should know because we're in a Zoom meeting right now. Next slide, please. The reason for COVID-19 success is due to its high likelihood for mutations. When a virus copies its genetic data, it does the job poorly, leading to many copies with slightly different traits, most of which are terrible, but some do lead to good mutations. While each mutation does take some time to emerge, every new prevalent variation will be more infectious than the last, the latest being the Delta variant, which the previous group also talked about. The data from the TMH FSU Rapid Response Lab in Leon County has shown that previously the dominant variant was the Alpha variant, which took 4 months to become dominant. However, now that variant's throne has been taken over by the Delta variant in only 1 month, it's predicted that the Delta variant will infect the majority of people. And while the Alpha variant made hospitalization due to COVID more likely, the Delta variant may almost double the chance to be hospitalized compared to the Alpha variant. It's hard to overstate the danger that comes from the Delta variant and why we need to learn what we can from the spread of the Alpha variant. Next slide, please. Therefore, our question that we will focus on is what can the spread of the previous variants tell us about the spread of the Delta variant, especially for places like FSU. We are trying to see what we can learn from Alpha so we can combat Delta. Next slide, please. Today we'll be discussing what exponential growth is, why the Delta variant is concerning, and how this affects universities and densely populated areas. And now to Blake, who will talk about exponential growth.
In order to understand spread, we must first understand exponential growth. Exponential growth increases its rate of growth proportionally to the size of the number. You can see things operate exponentially in everyday life, whether it's your compounding interest in your bank account or the spread of mold on an old loaf of bread. In some cases, the increasing growth rate can be helpful, but there are some scenarios in which it can cause things to get out of control. In the case of a forest fire, the spread covers a larger and larger area over time, and they often get to a point where efforts to stop it entirely fail because it has grown to a point where it just needs to run its course. How does this apply to COVID? COVID is much like a forest fire. As it spreads, the infected population exponentially increases, and as that number grows, it becomes less and less manageable, like a large-scale forest fire. When COVID first started in Wuhan, China, it was in a much more manageable state. It was possible to contain, and it could have run its short course like a small fire being snuffed out. Because it wasn't contained initially, we can see its exponential increase happening all around us, and it has become nearly impossible to cease all transmission purely because of the broad reach of the virus. Now Vishal will talk about the strain of importance.
So what makes the exponential spread of the Delta variant specifically concerning? To answer this, we have to look at a topic in epidemiology called R0, or the reproduction number. What this number tells you is the amount of people one person infected with the virus is expected to spread the virus to by the time they get better. So for the original virus in Wuhan, each person infected with the virus is expected to spread it to 2.4 to 2.6 different people by the time they get better. But for the alpha virus, each person is expected to spread it to 4 to 5 people by the time they get better. Now the Delta variant has an R0 much higher than the previous variants of 5 to 8. So each person infected with the Delta virus is expected to spread the virus to 5 to 8 different people by the time they get better. Now this is a visualization of how COVID spreads. It's focused on the original Wuhan virus with an R0 approximated to be 2. So this means that each person infected is expected to spread it to 2 different people. Now what this ring diagram tells us is that in the center, it shows us patient 0, the first person infected with the original virus. The first ring from the center is the amount of people that the person from the center infected the virus to. And the second ring from the center is the amount of people that the person in the first ring infected. So as you can see, after around 3 rounds of spread, the total number of people infected is around 15 for the Wuhan virus. Now for the Delta virus with an R0 approximated to be 6, after 3 rounds of spread, in total, about 259 people are infected. Now let's do the same experiment, except now we put some vaccinated people in the diagram. Now, the result of this is a significant drop in the amount of people infected. Next slide, please. This is shown in the blank sections of the ring diagram. And what this tells us is that vaccinations are critical at curbing the spread of future variants. We asked the question whether the spread of the alpha variant can be modeled exponentially. And to do this, we used a Python script, specifically matplotlib.pyplot, to analyze the data from Dr. Dennis's COVID-19 Rapid Response Lab at FSU. On the x-axis, we plotted the days from September 2020 to July of 2021. And on the y-axis, we put the cumulative occurrences of each variant observed. The Delta variant is plotted here in orange and the Alpha variant is plotted here in blue. Now, before Delta became apparent, the Alpha variant quickly became the dominant strain. And you can see it fits this exponential growth pattern from around April of 2021 to around June of 2021. But as soon as the Delta variant was observed in late June of 2021, it picks up immediately. It follows a similar exponential growth pattern to the Alpha variant, except it is only faster. This, in effect, is modeling evolution. All these strains are fighting in order to infect the most people, and only the most infectious will infect the most people and will therefore be observed the most. This next new dominant variant is Delta.
So with our previous slides in mind, we need to ask ourselves why it is important to understand the kinetics of COVID-19. One of the most pressing issues include the reopening of schools and future increases in cases. In fact, FSU and other universities are returning to a physical mode of learning with thousands of students stepping foot on campus after a year online. So what makes the spread of COVID-19 different at a large university? In many universities, lectures are held in high density enclosed spaces. In this picture alone, there are about 335 students all close together, tightly packed in an FSU lecture hall. So with COVID-19, we have to take into perspective how the number of unique contacts an individual has with other people can increase the risk of the disease spread. Let's take this student, for example. He has a full course load, which means that he has a full day of numerous unique contacts and exposures. With more classes, the number of unique contacts increases. On average, every person has 25 to 50 contacts per week, while the average student has about 150 to 200 contacts per week. To further view how vast unique contacts on a college campus are, let's take a look at a unique contact demo by Thomas Hoops, an FSU professor called Contactome. So here we are viewing the unique contacts of an example bio major at FSU. This student currently has 174 unique contacts. And what's cool about this application is that we can view each unique contact by class, and they're all color coded. So we can see which ones are the larger groups and which one are the smaller groups. And a very important thing that we need to take into perspective about COVID-19 is that we're not only dealing with this student's unique contacts, we're also dealing with this student's unique contacts and this student's unique contacts. This is especially important concerning COVID-19 because transmissibility occurs exponentially. And if one person gets infected on a university campus, it can very quickly lead to an outbreak among various other students and classes. Now let's continue our presentation with Carl.
So all in all, if we cannot control a virus's spread early, due to exponential growth, it will become difficult to manage like a fire spreading out of control. The earlier we put out or slow down this fire, the better, which is why the Delta variant is so concerning. Because it has a higher R0 than the previous virus variants, its spread will be even quicker, and without measures to stop or slow this, it will become very difficult to manage. On top of that, spreading within crowded places with many interactions between people, such as in universities, makes viral spread even faster and even more difficult to manage. Next slide, please. The Delta variant is the most infectious and dangerous variant so far due to these factors, and is predicted to spread to the majority of people who have not yet been vaccinated or exposed to a different variant. However, due to how much and how fast COVID mutates, it will not be the last variant either. One that is even more infectious can emerge. Our study was limited to only the people tested in Leon County, making it somewhat difficult to generalize some of our findings. Along with that, we were also limited by how much time and money we could put into the project, since our budget was effectively zero. Despite that, we do believe our findings can provide useful predictions about the next variant. The importance of studying how COVID spreads and finding ways to slow or prevent it cannot be overstated. Does anyone have any questions?
Thank you so much for another great, if a little disturbing presentation. Let's go ahead and thank them with our emojis and whatever we have at our disposal. And I will open the floor for questions. So great question in the chat. So first, thank you again for putting all this into context. The question is there a certain infection tipping point where you would recommend FSU to go digital? What kind of from your research, is there any sort of metric at which you would encourage institutions to make changes to their format of instruction?
I can take this one. So I don't know if you saw on the slides, but on one of them, it showed the exponential curve and like different sections of it labeled. So there was a less manageable, more manageable, and like not manageable at all section. And as soon as the spread of the variant hits like the exponential part of its growth, it becomes harder and harder to manage. And that's the stage where the Delta variant is right now. So yeah. So because of that spread, I think right now, if we go to in-person classes next year, then we'd have a huge portion of the unvaccinated people in FSU to be infected by these new variants, which is not what we want.
It's always challenging to connect the science to the policy. So thank you for attempting to answer that question, Vishal. Do we have any more questions? This was some fascinating homegrown data here, so I'm sure some of our Leon County residents might have questions. Dr. Heister.
So you were talking about possible generalizability of your results or challenges with that. So what would you say are some of the key features of Leon County or the population of Leon County that makes it different to say somewhere like New York or Miami or Wakulla County, which is south of Leon and not so populated?
I could take this question. Well, in like Florida, and especially like in like Leon County and stuff like that, there's definitely like pushback on like mask mandates. Like there's like confusion like on vaccination. And you saw in our PowerPoint how like and how a vaccinated individual really like affects the spread and how it limits it. So in New York, I'm sure like vaccination rates are significantly higher than here in Florida. So with the amount of vaccinated individuals, if we have more of those, then the spread would be greatly limited.
Thank you so much. That was a fascinating talk. Dr. Heister can introduce our next group.
So thank you again to our previous group for that fascinating, if not worrisome talk for those of us that live in Leon County. Our fourth group worked with structural biology data to better understand enzymes that may assist in the bioremediation of plastic waste, receiving mentorship and support from Dr. Beth Stroop. So take it away.
Okay. Hey, everybody. My name is Rahul Iyer.
Hi, I'm Julian Areco.
I'm Maggie Dykstra.
And I'm Sandhya Kumar, and our project is analyzing the unique properties of PETase in comparison to other PET degrading enzymes.
So as a quick overview, what is PET? PET is a molecule that stands for polyethylene terephthalate. It's the common plastic used for more durable items such as water bottles and polyester fibers. Because it is so durable, PET is extremely difficult to break down, which is becoming a concern for the environment as there is a lot of plastic accumulating in landfills. However, a discovery made from sludge samples near a Japanese PET water bottle plant in 2016 yielded a study of a bacteria called Ideonella sicyensis, which has the rare ability to degrade PET and convert it to energy with the use of an enzyme called PETase, which was the main subject of our research. So what is PETase? PETase is a PET degrading enzyme with unique capabilities. It has been shown to be the most effective in degrading PET out of all other enzymes that have the ability to do the task. And what makes it even more distinctive is that it can transform this detrimental plastic into its building blocks, which gives the golden opportunity to recycle plastic infinitely. Specifically, PETase degrades PET into BHET and MHET, which is further broken down to CO2 and energy. So we can see the mechanisms of PETase and PET in the diagram on the bottom right, and I'll pass it on to Maggie to discuss our questions and methods.
All right, so several enzymes have been identified as having PET degrading activity, most of which take the form of cutinase or lipase. However, PETase seems to be the most efficient and specific in its degradation of PET. In this project, we aim to answer the question, what makes PETase so unique in its degradation capabilities? In order to reach our conclusions, we first compared the polypeptide sequence of PETase with the sequence of several other PET degrading enzymes to determine which enzymes were most closely related to PETase using the tool ClustalOmega. We then analyzed the differences in the amino acid sequences which may contribute to the differences in PET degrading efficiency of closely related enzymes, specifically in the active site of PETase.
In order to identify what makes PETase unique, we wanted to find its close relatives, and then analyze what sections of the polypeptide are different and important. From there, we looked at a set of 18 sequenced PET degrading enzymes, and we chose 8 to analyze further, including PETase, as they produce similar byproducts of MHET and TPA. We used cholesterol omega to compare these enzymes and identified 2 other PET degrading enzymes that were closely related to PETase based on the cladogram. The 2 enzymes that we analyzed in addition to PETase were cutinase from Saccharomyces viridis and polyester hydrolase from Pseudomonas estes nigri. Given the similarity between these enzymes and PETase, we investigated the possibility that they are evolutionarily closely related.
So we wanted to see how structurally similar our proteins of interest were, and to do that, we used the match function in Chimera X to superimpose polyester hydrolase and cutinase on PETase. Structurally, they seem quite similar, as you can see from these videos. However, when comparing the polypeptide sequences, we began to notice more differences.
This figure compares the amino acid sequences of our 3 enzymes, so the polyester hydrolase, the PETase, and the cutinase respectively. The highlights indicate any similarities between 2 or more of the amino acids in the sequence. The dashes represent null information, a tool used by cholesterol omega in order to align the correct sections of the sequences. Towards the middle of the sequence, there are more similarities between the 3 strands, so in lines 4 and 5. But overall, it seems as though the polyester hydrolase and PETase enzymes were more similar. We also noticed that the beginning and ending segments did not see as many similarities between our 3 enzymes.
In a journal article exploring the active site of PETase, we were able to obtain vital information on how the active site is structured. This experiment used a ligand made of 4 MHET moieties, which are sections of a larger ligand. The first part of the active site we investigated was subsite 1, which has a deep and narrow shape that holds 1 of the 4 MHET moieties. Knowing the attributes of the active site and where the important amino acids were in the sequence, we looked how these amino acids were similar or different based on the results found in the cholesterol omega sequence from the previous slide. For subsite 1, we see no change in amino acids between the 3 enzymes, and thus no change in properties. However, we do know that in subsite 1 is vital for the secure binding of the ligand, specifically due to the amino acids methionine 161 and isoleucine 208, which both provide a hydrophobic surface at the bottom and side of the subsite, in addition to bonds created by tyrosine 87 and tryptophan 185, which stabilize the ligand for later degradation.
So we also looked at subsite 2. Subsite 2 of the PETase enzyme included 12 amino acids. It is longer and shallower than subsite 1, and it has 3 MHET moieties bound to it. It is divided into 3 smaller sections, subsites 2A, 2B, and 2C. Subsite 2 mainly interacts with these 3 MHET moieties through hydrophobic interactions, but polar interactions between the 3rd MHET moiety and serine 236 and asparagine 246 also occur. When comparing the properties of the amino acids in 236 and 246 for cutinase and polyester hydrolase, we found some differences. In the 236 amino acid, cutinase had threonine and polyester hydrolase had serine, meaning the properties were all conserved. However, in the 246 amino acid, cutinase was blank, and the polyester hydrolase had tyrosine, which is nonpolar and aromatic, as opposed to the polar and neutral asparagine present in PETase. It is important to note that most of the differences in amino acids occur in subsite 2, and so these differences may influence the overall difference in shape in the other sections.
The catalog triad is the 3rd section of the active site we examined, and it is largely responsible for PETase's hydrolytic properties. In PETase, it consists of serine 160, histidine 237, and aspartic acid 206. Serine donates electrons to help cleave PET, acting as a nucleophile, while histidine helps to polarize the serine molecule, and aspartic acid stabilizes the configuration when bound to PET. We noticed that these amino acids were strictly conserved in the two closely related relatives of PETase, suggesting that the catalytic triad may be largely responsible for the efficacy of PETase and other PET-degrading enzymes in PET degradation. Structurally, we noticed that the cutinase enzyme had a different arrangement of the catalytic triad in terms of differences between the three residues, which may also contribute to its lesser efficacy.
So we can reasonably conclude that one of the primary reasons for PETase's effectiveness is its amino acid properties that differ from that of other PET-degrading enzymes at certain locations. Since the sequences between different enzymes were conserved in the catalytic triad and subsite 1, we can speculate that these sequences are crucial to the PET degradation ability of these enzymes. The difference in efficiency, however, can be best attributed to subsite 2, where PETase had unique amino acids and properties at serine 238, arginine 280, and asparagine 246. We speculate that each of these amino acids play a crucial role in the efficacy of the catalytic activity of PETase. We can also extend the results of this investigation in the future by studying different aspects of the enzyme and the ligand, including the structure of the binding site and the chemical interactions between PETase and PET.
Although we were able to reach sound conclusions, there were a few limitations of our research. One limitation of our study is that we only focused on how the amino acid sequence of PETase differed from the other PET-degrading enzymes to determine efficiency. However, the inference that those amino acid changes are what caused efficacy changes could not be experimentally confirmed. In the future, we could induce mutations in these residues to determine if they truly impact PET degrading activity. Another limitation to note is the accuracy of the tools used. The ligand investigated in the journal that focused on the properties of the active site used X-ray crystallography, which is a tedious procedure that has a probability of error of the actual structure of the ligand. Clustal omega also has a small percentage, 2%, of inaccuracy, so the sequences may not have been perfectly aligned. One possible extension of this study, as we delve into a deeper study in the near future, would be studying how the strength of the bonds between the ligand and PETase change based on the amino acid alterations seen here in Chimera X, as mentioned earlier.
So here are some of the sources that we used in our investigation. And moving on to the next slide, we would like to acknowledge Dr. Stroop, Camille Luna, Dr. Staling, and our TAs for all of their help. And in addition, we would like to open the floor to any questions.
Thank you so much for another fantastic presentation. That was fascinating. So let's go ahead and thank them again with the emojis. And then also I'm ready to take any questions. And I'll just say, I know there's a couple former YSP students out there, some alumni, and I'd love an alumni question at some point if you have one. So let's see. Okay, we have one question in the chat in the meantime. So how do you think knowledge of PETase might be helpful in dealing with our microplastic situation in the world's ocean? Can you see how this would be applied to that problem?
I can take that question. So definitely there's a huge possibility of PETase acting in bioremediation, including in systems like the ocean. It is unique to this one bacteria, Ideonella saccharinosus, which would need to be tested for how it grows in the ocean and its ability to live in that environment, maybe not acting as an invasive species, which would be another issue. But I do know that some aspects of bioremediation for this enzyme include sort of a cocktail of this enzyme and also other sort of buffer organisms that could help to control its growth and allow for controlled degradation of plastics.
Thank you so much for the thorough answer. We do have time for another question if anyone else has a question. Oh boy, yay, Lindsay Yang, go ahead.
Yeah, so with this information about why PETase is more effective than other enzymes, do you see it being possible in the future to maybe engineer other more effective enzymes to degrade PET that's not just PETase?
Yeah, I can go ahead and take that question. I believe that was kind of part of our motivation, was that we recognize the plastics being such a big issue for the environment. that we wanted to investigate what made this enzyme unique in order to try and see, or to kind of investigate a way that we could use that in other enzymes, like specifically the configuration of the active site, what makes that unique, so that maybe we could implement that for other enzymes and biomedically or bioengineer that to be in different organisms, not necessarily just the Ideonellus psychiensis.
Got it. Thank you. That was a really good presentation. Thank you for it, guys.
Awesome. Thank you so much. Okay, well, in the interest of time, we do have to move it along. I'll say we are right on schedule. We're at the halfway point at this point, and I will pass it off to Dr. Heister to introduce the next group.
Great. Thank you, group. So the previous group, and again, for thinking about those real world implications, which were the challenges that we're facing. So our fifth group also worked with structural biology data, in this case, exploring enzymes with potential applications in medical bioremediation of cholesterol. This group also received mentorship and support from Dr. Beth Stroop. So take it away.
Hi, everyone. Our project was on comparing the efficiency of 3-ketosteroid delta-1 dehydrogenase and cholesterol oxidase at degrading 7-ketocholesterol. I'm Brighton Jiang.
I'm Surbi Kumar.
I'm Anuj Prajapati.
I'm Rishi Shah.
So bioremediation in itself is using natural organisms such as bacteria to clean up polluted areas. Medical bioremediation in this context is a term coined to describe the use of enzymes from specific bacteria to clean up plaques, or plaques, which are the pollutants in this case, that are blocking normal functioning of the human body. This pollutant is caused by 7-ketocholesterol, also known as 7-KC, which is a product of the oxidation of cholesterol. When accumulated, 7-KC produces plaques and toxicity that leads to aging diseases such as Alzheimer's and atherosclerosis. A medical bioremediation approach uses unique enzymes from bacteria that have been shown to degrade 7-KC.
Many organisms and bacteria have proven to be able to catabolize 7-KC. We initially looked at a specific chromobacterium, which is proven to degrade 7-KC using a cholesterol oxidase enzyme. We used NCBI's BLAST tool to create an evolutionary tree which identified similar bacteria based on sequence relatedness to the gene that encoded for cholesterol oxidase. We noticed that Rhodococcus erythropolis, shown by the arrow, was also in the tree and that it had an 89% relatedness to the chromobacterium. Literature predicts that under optimum conditions, Rhodococcus erythropolis was found to degrade 93% of 7-KC within 15 days of incubation. We then found that the enzyme in Rhodococcus that was capable of degrading 7-KC was 3-ketosteroid delta-1 dehydrogenase. So we framed our project on comparing the efficiency of these two enzymes.
So the purpose of the study was to further understand and compare the efficiency of cholesterol oxidase in Chromobacterium and 3-ketosteroid delta-1 dehydrogenase in Rhodococcus erythropolis to degrade 7-KC by looking at and comparing the binding affinity and Gibbs free energy.
Our hypothesis is that if we evaluate the efficiency of both cholesterol oxidase and 3-ketosteroid delta-1 dehydrogenase at degrading 7-ketocholesterol, or 7-KC, then we expect that cholesterol oxidase will have a greater efficiency at degrading 7-KC because it has more hydrophobic interactions with 7-KC than 3-ketosteroid delta-1 dehydrogenase.
In order to accomplish this, we obtained the amino acid sequence of cholesterol oxidase in 3-ketosteroid delta-1 dehydrogenase from the protein data bank. In order to create a full 3D model of these enzymes, which was not available to us, we utilized a threading algorithm provided by FireSquared to predict these structures. We then found a model of the ligand 7-KC to use in our models. Next, we imported the enzymes and the ligand into Chimera X, where we were able to use Chimera X matchmaker function tool to align 7-KC to the active sites of the two bacterial enzymes, 3-ketosteroid delta-1 dehydrogenase and cholesterol oxidase. We then used a docking program to identify the active sites of both enzymes that would minimize the activation energy of the enzymatic reaction and would thus be the most ideal site for 7-KC to bind to. Here on the left in figure 5, you can see 7-KC in red bound to 3-ketosteroid delta-1 dehydrogenase, which is in blue. In figure 6, you can see 7-KC, which is in pink, bound to cholesterol oxidase, which is in gold. Both of these models were created after being put through the Swiss docking program and generated in Chimera X. To compare the efficiencies of these enzymes, we utilized the metrics of binding affinity and Gibbs free energy, some of the only data available to us. Using a neural network software, we were able to calculate the predicted binding affinities and Gibbs free energies of both our models given the current protein sequence. Binding affinity is the strength of the binding interaction between a single biomolecule, such as protein or DNA, to its ligand or binding partner, such as a drug or an inhibitor. Binding affinity is typically measured and reported by the equilibrium dissociation constant, or KD, which is used to evaluate and rank order strengths of biomolecular interactions. The higher the value of PKD, the greater the binding affinity of the ligand for its target. Knowing this, we can see that the binding affinity, PKD, of 3-ketosteroid delta-1 dehydrogenase for both its monomer site and polymer site are less than the binding affinity for cholesterol oxidase and 7-KC. The high standard deviations here on this chart could be due to the error that comes from the predicted model generated by the threading algorithm that we used. The Gibbs free energy value is a good indicator for the spontaneity of a reaction, with lower delta G value signifying a more spontaneous reaction. Although delta G does not directly affect enzyme efficiency, it can indicate that a reaction occurs spontaneously. This means an enzymatic reaction is favored, but not necessarily more efficient. The delta G value for cholesterol oxidase is less than that of both the monomer and polymer of 3-ketosteroid delta-1 dehydrogenase, indicating a more spontaneous process. Again, this means that the enzymatic reaction between 7-KC and cholesterol oxidase could occur more spontaneously, but this is not always the case.
Figure 7 is the molecular lipophilicity potential model of Rhodococcus erythropolis 3-ketosteroid delta-1 dehydrogenase and 7-KC. Since we are doing with a steroid, the only interactions that are going to take place between the enzyme and ligand are hydrogen bonding and hydrophobic interactions. With steroids being hydrophobic, they are more likely to attract to the hydrophobic parts of the enzyme. We can use this property to narrow down our gene mutations to only genes that are hydrophobic. This means that when we make these mutations, the binding affinity will change and not stay stagnant, which benefits our analysis. In figure 7, the gold is more hydrophobic. When analyzing the model for cholesterol oxidase from Chromobacterium, it comes to be clear that there are more gold spots than the other enzyme. This means that there are more hydrophobic interactions within this enzyme and the ligand, which leads to higher binding affinity and possibly efficiency.
Because 7-KC is a lipid, it is more likely to bind better when the binding site is hydrophobic. We changed 8 of the original amino acids on the enzyme cholesterol oxidase to tryptophan, a hydrophobic amino acid that frequently appeared in hydrophobic areas where 7-KC binded. We found that changing the active site to a more hydrophobic area increased the binding affinity of cholesterol oxidase and lowered the delta G value. We can see that there are the 8 amino acids that we changed created a more hydrophobic area, and you can see that by the increased yellow area in the gold. And it is important to note that although we see a higher binding affinity, we don't know if these mutations we made continue to allow the enzyme to carry out its normal functioning, as they were not chosen selectively. And this has to be tested to safely conclude that there is in fact a positive effect of the mutations on enzyme functioning.
So by comparing the binding affinities using PKD of the two enzymes, we can find the one that is possibly more efficient at degrading 7-KC and thereby further improve the binding affinity by mutating the enzyme to be more hydrophobic. Future research would involve experimental testing utilizing this enzyme and its ability to be inserted, degrade, and figuring out how to get it past the blood-brain barrier. Eventually, this would pose a new potential preventative treatment to avoid and combat the aging-related diseases such as atherosclerosis, Alzheimer's, and other neurodegenerative diseases that result due to the buildup of plaques in 7-KC and break them down into substances that can be naturally disposed of. This would eventually lead to significant steps amongst these illnesses, especially since these plaques are resistant to cellular catabolism and are hard to degrade, which is what makes these diseases so difficult to treat. Moreover, it would provide a lot more information into this niche area of bioremediation that is yet to be further explored.
Although the conclusions are exciting, we have to note some limitations. Because medical bioremediation is less studied than other types of bioremediation, a lot of the available papers and data we looked at were done in the early 2000s. This limited some of the options we had. We weren't able to find data on some of the enzymes we wanted to study and couldn't study as many enzymes as we would have liked. For example, we had to find the active site of 7kc ourselves instead of using published information. We assume that the active site that the ligand binds to is the place with the smallest Gibbs free energy. While logical, this might not be true because binding is such a complex topic and other factors can influence the site. Additionally, when mutating the enzyme, we didn't change the active site, when it might have changed along with the mutation. When selecting mutations to change, we mutated all the amino acids to tryptophan, which would be unlikely to happen in a real environment. Also, the two enzymes that we looked at might have cofactors that would be affected, which we didn't look at either. We assumed that a tighter bind between the enzyme and ligand resulted in a more efficient reaction. While the higher mutated binding affinities could suggest a more efficient degradation, possibly the opposite could be true, where a too tight of a bind could inhibit the enzyme. Our results would have to be tested experimentally in a controlled environment to conclusively see the positive effect of our mutations. We'd like to especially thank Dr. Stroop and Luna for all their help and guidance during our project. Are there any questions?
Thank you so much for another great presentation. Let's give them another round of applause of celebratory emojis and whatnot while we wait for some questions. Looking for any hands raised. So we do have one question in the chat from Barb Shoplock. It is, how did your group decide on this interesting topic? Will you ever think of cholesterol the same?
I can take this question. So when we were looking at the topic of bioremediation, we wanted to get more information about what exactly it is and what the applications are. And we saw a lot of environmental applications, but then we saw this medical aspect about bioremediation that kind of intrigued our eyes. So that's why we wanted to pursue it. And that's kind of what led us to choosing this topic. And we probably will not look at cholesterol the same.
See, we do have a hand raised. Dr. Heister, do you have a question?
So I'm curious, you said there was kind of a dearth of literature. There was only stuff in the early 2000s. So do you have any feel for why this enzyme went out of fashion or wasn't studied so hard? Why is it so niche?
I can take this question. I think because like the area of medical bioremediation is just so it's so complex, because especially one of the major applications would be to treat neurodegenerative disorders that are caused by the buildup of plaques of 7kc. But one of the major challenges I think Brighton also mentioned was, could we even theoretically get an enzyme that could be created from from Chromobacterium cholesterol oxidase or such? Could we get that past like the blood-brain barrier that's so selective? And if it could even be used as a potential treatment. So I think a lot of the research that hasn't happened in so long is because of trying to figure out if we could even get it through past the blood-brain barrier as a potential treatment.
Fascinating. Okay, we can take one more question. It looks like Sanjay Kumar has a question.
Yeah, I was just wondering if there was a reason that you mutated those 8 amino acids to tryptophan as opposed to maybe other nonpolar amino acids?
I can take this question. So when we were looking at the enzyme cholesterol oxidase in Chimera X, we had seen like some areas of yellow, which yellow indicates it's more hydrophobic. And in those areas that were nearby where 7kc is binding, the main amino acid is tryptophan. And tryptophan is a more hydrophobic amino acid than the 8 amino acids that we mutated. So we decided to mutate it all to tryptophan. And obviously this is not necessarily ideal because it could change some of the structural properties of the enzyme, but we thought that that might be a good starting point for our mutations of the enzyme. And we did see that it did increase the binding affinity, which was very interesting.
Thank you so much again. That's all the time we have for questions. So I'll turn it back to Dr. Heister to introduce the next group.
Great. I'll reiterate saying thank you again to our previous group. So our next and 6th group explored the molecular dynamics of the novel coronavirus, receiving mentorship and support from Professor Wei Yang's lab, especially Dr. Zheng, Dr. Wu, and Tianming Chu.
Hello, everyone. My name is Henry Chen, and my fellow group members are Margarita Krylova, John Wu, Audrey Lee, and Olivia Tang. And together, we researched the topic of molecular dynamic simulation discovery of cryptic sites in SARS-CoV-2 main protease. Our research was done under the mentorship of Dr. Wei Yang, Dr. Liangqing Zhang, Dr. Dongsheng Wu, and Tianmin Qiu.
So to introduce the topic of SARS-CoV-2 Mpro, due to the spread of COVID-19 pandemic, the SARS-CoV-2 main protease, which is an enzyme, caught a large amount of attention. This enzyme is essential to the life cycle of the COVID-19 virus and it is the first SARS-CoV-2 protein whose structure was able to be identified and recorded in the protein structure database. The molecule is a homodimer and consists of two polypeptide chains that are identical in the order, number, and kind of the amino acid residues. The understanding of the SARS-CoV-2 Mpro structure can lead to the identification of the cryptic sites of the protein. Cryptic sites form pockets in ligand-bound structures, but whenever the structures are unbound, the cryptic sites are absent or occluded. Finding cryptic sites is important for the development of a drug to combat the pathogen, since cryptic pockets can function as druggable sites.
So let's first understand the basics. As cryptic sites are areas that drugs can bind to, they are extremely important in medical research, especially when dealing with new diseases such as the coronavirus, and there is a need to find an effective drug already on the market. There are two ways cryptic sites can form.
When following the conformational selection mechanism, the conformational change occurs before the binding of a ligand, making the formation of cryptic pockets predictable. These cryptic sites are good drug targets due to the large pocket size. On the other hand, the conformational change of the induced fit model occurs after the ligand binding, making the cryptic site formation unpredictable unless the ligands are specified. These pockets have limited druggability due to the limited cavity size. Therefore, we use the conformational selection mechanism in our simulation.
Now for our methodology. Our central research question is, how can we use MD simulations to identify conformational change of the SARS-CoV-2 main protease and evaluate the possibility of cryptic site occurrence? And our goal is to use MD simulation to determine sites of conformational change, as well as the possibility of cryptic formation within the conformed sites.
Throughout the research project, we used 5 technical tools that largely helped us to run the simulations and collect the data. The first technical tool that we used was Visual Molecular Dynamics, also known as VMD. We used that tool to import and visualize the data. After, we used Linux to connect to the FSU's High Performance Computing or HPC cluster to run the simulations. We also used the Molecular Dynamics or MD simulation to perform molecular simulations. CharmGUI was used to create simulation protocols and environmental conditions for the proteins, such as water box size and types of ions. Finally, we used RCSB protein data bank to find the PDB files for the needed proteins.
We first found the 6LU7 protein in the RCSB protein data bank, imported the PDB file of the protein into CharmGUI, and copied the file produced into our high-performance computing system. Next, we ran our MD simulation and checked our results after a few days. Using XMGrace, we were able to plot and scale both lambda and root mean square displacement values and compare them with time and free energy values. Finally, we created crystal structure animations using VMD to analyze conformational change shown on the graphs.
Now let's show our data and data analysis. The molecular dynamics simulation outputted thousands of data points detailing the lambda step value, the calculation of free energy at that specific value, and also the change in free energy with respect to time. A screenshot of our outputted data is shown on the right. Free energy is defined as the energy available in the protein system that is available to do useful work. It is crucial for identifying conformational changes because free energy correlates with the activities levels within the protein, which will eventually impact the conformational change. For example, when bonds in the protein break and form, it all alters the free energy values. We also used root mean square displacement, or RMSD, as a measure of the deviation of the position over time with respect to the initial reference frame of the protein. Since the trajectories outputted by the MD simulation did not contain RMSD, we had to scale it ourselves using a calculated conversion factor shown on the right. Here we have the free energy landscape graphs. On the y-axis of both graphs, we have the free energy measured in kilocalories per mole, and on the x-axis of the graphs, we have the lambda value and the RMSD scaled value measured in angstroms. The major features of both graphs are the 2 local minima. Throughout the entire simulation landscape, the protein is in constant dynamic conformational change. However, at these specific free energy minimums, they show the conformations where the protein is relatively stable, which is why we are specifically focusing on them. We were primarily focused on the second minimum, located at around 1.8 angstroms, because the first minimum shown likely corresponds with the protein achieving equilibrium with the surrounding salt solution rather than a potential cryptic site. Here we have the time graphs. On the y-axis of these graphs, we have lambda and RMSD, and on the x-axis of the graphs, we have time measured in nanoseconds. Originally, the MD simulation outputted the time in femtoseconds, so we had to convert it to nanoseconds. From the graph, we can see many dips in RMSD. Most notably, there is a dip at around 10 nanoseconds. The RMSD dips here from down to around 1.8 angstroms, which is exactly what we observed from the previous graphs. Therefore, it is safe to assume that this dip in the RMSD versus time graph corresponds with the conformational change from before. Next, Margarita will show the animation of the protein dynamics at 10 nanoseconds to show this conformational change.
Here we're able to see the two crystal structures. The one on the left is the first frame of the simulation, or the initial protein. The structure on the right is the last frame, frame 1734. The structure has 4 predicted cryptic sites, which are circled. When we compare each of the sites, we can see that the active site experienced the most change. The distal site had some change, and the dimer interface region experienced the least change. Since the molecule is symmetrical, the same phenomena is observed on both sides. If we look at the active site of the molecule, it is evident that there is a major conformational change, which corresponds to the minimas on the graphs discussed by Henry. The most change can be seen on the left side of the molecule, since the cryptic pocket opening there is the largest. We can also see some change at the distal region. However, the dimer interface mainly stays unchangeable. On this slide, the alignment combines the structure of the first and the last frames to visualize the conformational change and make it much easier to see. The structure supports the fact that the most structural change is seen at the active site of the molecule. Here we can also see that for the most part, the structure did not experience any major changes. For example, the circled sign loops had some minor changes. However, those changes were not enough to impact the pocket opening. The alpha helices experienced some major changes. If we look at the initial structure marked orange, we can see that it is proximal to the sign loops. However, the final conformed protein, which is purple, is farther away from the sign loops, resulting in an opening and formation of a cryptic pocket.
Our conclusions were the following. We met both of our initial goals as we were able to identify points of conformational change using the free energy graph and also evaluate their likeness of being a cryptic site by using frame-by-frame analysis and animation. Our data results suggest that there was a conformational change at around RMSD equals 1.8 angstroms. Analyzing the animation at the same time corresponding to the conformational change around 10 nanoseconds, we saw clear indications of a site opening at the region labeled active site. Tara Stein et al. Also noted similar findings at that region, so we conclude that the conformational change at 1.8 angstroms is likely a predicted cryptic site.
Regarding limitations, the first one is the time constraint, which meant that the MD simulation did not have enough time to converge, so then we couldn't get the entire protein free energy landscape. And currently the simulation is still running and collecting more data. Next, as a result of the time constraint, we have limited data to work with, and these limitations could possibly be eliminated if we just have more time to wait for the simulation to finish running, as then we could collect more data and extend our research. However, due to the our results were only able to go to the extent of establishing some possibility that cryptic sites formed.
The research we conducted has a lot of potential and can be expanded in a number of ways. If the MD simulation is allowed to run for a longer period of time, more data can be collected and the research can be repeated, possibly yielding better results. In addition, local minima in relation to conformational properties of the animation could be studied in more detail. Finally, properties of each cryptic site and how they interact with a given drug could be studied to possibly discover a drug that could combat the pathogen.
We would like to once again thank our mentors for guiding us through our research. We'll open the floor for any questions now.
Thank you so much for another great presentation. Again, I would love to get questions from any alumni who are still in the audience or anyone else. And let's, in the meantime, while you prepare your questions, give them some more plaudits here. Awesome. Okay. So there's no external questions. We do have a question from Barb Shoplock in the chat. So it says, thank you for all the background info to help us understand your research topic. What do you think is the most useful finding from your research? So if you were being funded, what do you think would be the most productive area of focus?
I can take that question. I think that the most important structural change that we saw was at the active side, which was on the left side of the molecule, since we saw the most of the opening there. So in case if we were funded, I guess that would be the most important aspect to focus on in the molecule.
And to build off that response, since we saw the opening, this could be used to fund further research on drug development that specifically targets that predicted cryptic site, as well as fund more additional research on the structure of that predicted cryptic site.
Thank you so much. I have another question in a private chat. So as your presentation stated, a limitation is that your simulation is still running. So do you think that it'll give you the result you desire? How much time do you think until it's completed? What kind of surprises might you encounter when you do finally get the result?
I can take this one. So the simulation is still running and it may take a couple weeks for it to fully converge. So what we expect is for more of these minimums to occur on the free energy versus RMSD graphs, which could indicate more areas to study in the animation so we can see more openings and closings. Because right now we only had this significant opening at 1.8 angstroms, but I believe if we run the simulation longer, there should be more instances of this.
Great, thank you so much again. Because we are short on time, I'm going to turn it back over to Dr. Heister to introduce our last two groups.
Great. So thank you again to our previous group, and I hope you eventually get the answers from the supercomputer. So our 7th and 8th groups both investigated data from tank experiments with applications in pollution dynamics, working in collaboration with the FSU Geophysical Fluid Dynamics Institute and the Department of Scientific Computing, receiving support and mentorship from Dr. Catherine Hancock, Ashley Gannon, Darren Sagal, and Jamaica Greenwood.
Hi, so my name is Kritika Subramanian, and my group Ankita Nath, Abhishek Suresh, Kenneth CS, and Tushar Gona will be presenting our research on an analysis of building arrangements in urban settings and its effects on pollution dispersion. Air pollution is a very large problem as it affects many people around the world. Specifically in Florida, air pollution becomes a major problem due to the close proximity of the Everglades because of the forest fires that can lead to smoke dispersion to the surrounding areas, according to USGS in 2015. Not only does smoke dispersion cause a problem, but so does the devastating hurricanes that happen yearly here. According to the Fish and Wildlife Foundation of Florida, hurricanes can destroy energy and chemical production facilities, gas stations, and other businesses that release toxic chemicals and materials into the environment, which further causes more pollution. And lastly, natural phenomena and disasters can pick up pollutant deposits that have gone untouched for years, reintroducing them back into the ecosystem and the environment. So specifically, how has air pollution affected the atmosphere for the past few years? Overall, NASA in 2021 has stated that there was a decrease in air pollution from the years 1998 to 2012 in the United States. However, due to more emission concentrations, the air pollution indexes have changed from year to year. This further can be seen in the graph provided by the New York Times, where new data has revealed that air pollution has actually been increasing since 2016, reversing previous trends towards cleaner air.
Our research question states, how does the proximity of buildings to each other affect pollution dispersion in an urban setting? To answer this question, we hypothesize that if there are fewer buildings in close proximity in an urban setting, then pollution dispersion will be minimized at this position, while highly dense urban areas will maximize pollution dispersion.
So to solve this question, we decided to model the pollution dispersion against different building configurations and different fluid speeds. The different building configurations models different environments in which air pollution will be present, and the different fluid speeds model the different possible wind speeds for those environments. No obstructions represents a rural environment. 1 obstruction represents an urban environment, and 4 obstructions represents a dense urban environment like a city. So we processed plume video data from FSU's Geophysical Fluid Dynamics Laboratory. For every pixel in every frame of the video, we determined whether the pixel had a dark, medium, or light shade. From this, we determined what percentage of the screen each shade consisted of. We did this all with a homebrewed custom Python program. The data was then plotted to show the trends, along with the standard population deviation value for each percentage.
We have 2 independent variables, the number of buildings that are being simulated in the water tank, along with the different liquid flow rates that simulate the intensity of smoke plumes. The different flow rates represent the intensity and speed in which the pollution plume could possibly take on. We then measure how the pollutant interacts with the various building structures around the plume in different scenarios. Dr. Hancock and a team of graduate students assisted us in acquiring video footage of this phenomenon by creating multiple experiments that used water flow rates and building structures, including no obstruction, which was 0 buildings, 1 building, and 4 buildings. They first introduced the plume in question, a mixture of rubbing alcohol and red dye, into the water tank by way of a pipeline that leads underneath the flooring of the tank, shown by the arrow-labeled plume flow. This plume would then be exposed to the open water, representing the clean air of our urban setting, and any obstructions that may or may not follow it. Laminar water flow, simulating the movement of wind, acted as a background flow that carried this plume. This can be seen in the photo where the laminar flow is moving from the right to the left through the tank. To result in a laminar flow, the Reynolds number would have to have a higher viscous force. The Reynolds number allows us to predict flow patterns in the different fluid flow situations, such as this one. The video of this interaction between the plume and the structures is then recorded for our analysis. The picture on the left is a screen grab of a frame that is unobstructed. We can clearly see the direct path on which the plume seems to take. The picture in the middle shows 1 building obstruction, our minimum. This screen grab shows the plume being dispersed in a completely different manner. The 4 building obstruction, our maximum, shows a dispersion pattern similar to the no obstructions, which is interesting to note. Also in these pictures are different shades of the red dye. We can see light, medium, and dark areas of the plume. These different areas reflect each other because the dark areas dissipates into the medium concentration, which then dissipates into the low light concentration. This phenomenon is observed over time, which will be measured in frames. It is a foundation on which our analysis is based off of.
So we obtained data with no obstructions for each of the 3 water floor plume rates. The first impression of this data is that there was not much variation among the pixel count percentages for either the light and medium pixel colors. This indicates to us that this is a lack of variation and as a result, a lesser degree of pollution dispersion. This can be explained by the fluid's laminar flow, which at no point is interrupted. This leads to relatively little mixing in the flow and can possibly lead to pockets of pollutants being created. Here we have the results for when there was 1 obstruction, which was our data for 1 building. For the plume rates of 1 meters per second and 4 meters per second, the pixel count percentage for the light and medium colors fluctuated or varied constantly throughout the frame rates. While the fluctuation for when the plume rate at 15 meters per second was not extreme as the previous representations, the turbulent flow that is occurring as a result of impact with the 1 obstruction has led to a high level of pollution dispersion. For our data with 4 obstructions, which is represented by 4 buildings, we see our data revert back to trends that we identified for no obstructions. There's a low level of variation that is similar to the data of no obstructions, which indicates that at this point, laminar flow is occurring and there is low pollution dispersion. This again may also indicate that pollutants are getting clustered into pockets. And one significant trend throughout the data for all 3 models, however, at a much higher rate for the one obstruction data, is that medium and light data reflect each other, which is due to the medium concentration dissipating into the light concentration. So for each of the dark, medium, and light pixel percentages among the videos that were analyzed, we calculated the standard population deviation values for each. For instance, at 1 meters per second for 1 obstruction, we identify a population standard deviation of 3.86 for the medium shade and 3.87 for the light shade. In order to get a clearer view on the trends of these values, we will look into the average of these deviations. So looking into our average standard deviations, it is clearly seen that between the standard deviations for the light and medium shade at 1 obstructions is significantly higher than both the corresponding values for both no obstructions and 4 obstructions. This is a clear indication that the values of this range are spread out over a relatively wide range and therefore a high level of pollution dispersion. Contrarily, while the data for 4 obstructions was slightly more varied than that of no obstructions, they are both around the range of 1 to 2 for the light and medium shades. So as we increase the number of buildings that are at the site of the source of pollution, the dispersion of pollutants decreases. This also may indicate a shift from turbulent to laminar flow at the transition from one obstruction to 4 obstructions.
From the data, we observed that the no obstruction arrangement resulted in the lowest amount of pollution dispersion, whereas the one building arrangement resulted in the highest dispersion. What didn't support our hypothesis, though, is that the 4 building arrangement was similar in dispersion to the no obstruction arrangement and was way less than the 1 building arrangement. So from this, we like to interpret this data as saying that as more obstructions are added, the flume will approach laminar flow, as well as the dispersion rates of the unobstructed data. And from these graphs, as you can see, the light and medium, we'll focus on the light and medium lines for each of them. No obstruction, 1 obstruction, and 4 obstructions. As you can see, the graphs for no obstruction and 4 obstructions are more similar, as there's less erratic movements in the graph of the light and mediums. However, in 1 obstruction, there's a lot of variation, which means that the particles are interacting with each other more and transferring back and forth into pockets of concentration and pockets that are less concentrated. From this, we can infer that if you add more buildings, as the particles will begin to interact less with each other and result in closer to a laminar flow that was experienced at the beginning with no obstructions. Some limitations we had was the limited amount of data. We only had data for no obstruction, 1 obstruction, and 4 obstructions. However, having data for more obstructions would help us solidify our conclusions. If we had maybe 2 obstructions or 6 obstructions and somewhere in between, where you could find a general trend for more values and help solidify it. Also, we can't change the diversity of the obstructions. Right now, we looked at every obstruction being the same, but in a real city, obstructions are different shapes, buildings are never the same, and that might influence the results in a real world scenario. Also, with the flume machine, there might be some confounding variables that affected our data. So in the future, we can try testing with different environmental factors, like maybe how temperature and the wind patterns of the fluids affect how they disperse. We could also try with different parameters, for example, like changing the densities of the dye that's used, etc., in order to see its effect on dispersion. And further, we can try to run correlation tests with real world data in order to see how exactly they compare.
So we would like to thank Dr. Hancock and her team of graduate students, Ms. Ashley Gannon, Ms. Darren Sagal, and Ms. Jamaica Greenwood for everything that they've done in assisting us with our project. We'd also like to thank Dr. Staling and all the other professors and counselors who were involved in the program and helped us get our project off the ground and running. Thank you.
Thank you so much. So let's go ahead and give them another round of applause here. And I will open the floor to questions. I have one if no one else does. Okay, it looks like we have a question from Dr. Heister.
I'm happy for you to go ahead and ask your question first, Dr. Staling, as you haven't really had a chance yet today.
Sure, okay, I'll ask my question. So my question was kind of, when you're doing these flume simulations, there's sort of like this transient response, and then I'd imagine eventually you kind of get it equilibrated, like when you're first introducing the pollutant. And I was wondering what you thought is more relevant to what types of real world situations. Is it like that spin up of what's first happening, or is there some kind of, are the equilibria reached in the different scenarios different? I'm just curious if and how you would consider that.
Okay, I'll answer this question. So we started recording the data when the plume was already in equilibrium. We didn't record any data starting at the beginning, just because we wanted to measure it in a point of equilibrium.
Awesome. Okay. We do have another question in the chat from Nicholas. Are there any alternative sustainable city designs you would like to experiment with to examine the impact on pollution levels?
Yeah, I can answer this. So for an urban planner using this like information, they can normally power plants or other sources of pollution, they either have no buildings around them or like a lot of buildings clustered around them. But rather, if it's like sparsely populated with like small buildings every here and there, I feel from our results, that would be the most dispersion, meaning that the pollutant wouldn't concentrate in one area and affect one group of people, but rather quickly dissipate and have less of an effect overall.
I'd like to add on to that answer. So we looked at the laminar flow coming in from the right to left and the building being obstructed by that and the flume flowing to the right to the left. So if we were to experiment on like this suitable city designs, maybe we could like get some wind data or something similar that we can like cross-reference the building. So like the laminar flow is going in the opposite direction of the wind flow. So the wind carries the plume away from the city and the population rather than spreading it all over the city.
So I have a couple of questions in chat and private chat that I'm going to try to combine into one last question, which is, so in this study, you were mostly looking at number of buildings. To what extent do you think other potential factors might be important, such as the spacing between the buildings or their orientation? Do you have any feel for that and how big of a factor those things might be?
Yeah, I can take this question. So yeah, to like a very high extent that the orientation of these buildings and the spacing would affect our results and like affect the dispersion of the pollutants. Well, and just because when we were doing our research and the data that we were using, we did with 4 identical buildings. So it was hard for us to visualize like how it would be done with actual city designs. But if we were to do that, we would be able to account for, like, say, if there's a hole like in the top of a building where the pollutants can go through, or there's like a lot of space or there's different spacing, because also in our data, all the buildings were spaced, like for the 4 building obstructions at least, they were all spaced like at the same, like in the same square, like at the same distance from each other. So yeah, it would definitely affect it.
Awesome, thank you so much. In the interest of time, we do have to move along. So Dr. Heister, if you would introduce our last group.
Great, thank you to the previous group. My question was also about orientation, so I appreciate you answering that and other people asking it too. So our final and 8th group also worked with experimental fluid dynamics data and drew comparisons with real-world data to understand how wind speed affects pollution dispersal. So go ahead, group 8.
Thank you for your introduction, Dr. Heister. And thank you to everyone who has stuck until the end to hear our presentation. My name is Leonard Volchek, and today Dylan Epstein-Gross, Krishna Patel, Anna Lu, Prabhas Kurapati, and I will be introducing you to the results we arrived at by analyzing the relationship between wind speed and atmospheric pollution concentration using experimental and real-world data. So although as of present, the atmospheric pollution concentrations are below the national average, many recent fluctuations in natural conditions, more frequent natural disasters, and the industry expanding, the air quality has seen a decline, which may detrimentally affect the health of populations. Recent studies have also shown both the speeding up and slowing down of wind in certain regions of the world due to both natural climate cycles and global warming. Taking this into account, our group decided to investigate the relationship between wind speed and atmospheric pollution concentration using video data of a pollution dispersion simulation in a water flume provided to us by FSU's Geophysical Fluid Dynamics Institute. Though previous studies have established the existence of an inverse relationship between wind speed and atmospheric pollution concentration, few studies have implemented a visualization by using a water flume. A further confirmation of this relationship using this flume data would signify that in the near future, we must watch for pollution concentrations increasing in the parts of the world undergoing wind stilling, and as a result, an increase in cases of respiratory problems. Additionally, despite the fact that work has been done analyzing the general relationship, the studies mostly took into account individual cities and specific neighborhoods. For example, an unnamed neighborhood in central London, UK, studied by Arstadami and her team. In order to add on to this research, we determined that we wanted to also inspect the wind speed to pollution concentration correlation on a broader scale, sampling AQI and wind speed data from 3 pairs of climatically similar US cities and looking at a comparison between the flume analysis results and the city pair analysis results. The research question we posed was, how is ground level atmospheric pollution concentration affected by wind speed? To which we hypothesized that with higher wind speeds, the pollution concentration will be lower, and with lower wind speeds, the pollution concentration will be higher.
To start off with testing this hypothesis, we decided to use data from a real physical experiment run by the FSU Geophysical Fluid Dynamics Institute, While most studies of air pollution patterns rely on data from computer simulations or large-scale observations, a water tank experiment like this one can have important advantages. As Rudin et al. Puts it, a carefully designed tank can be a good scale model of real topography and realistic atmospheric conditions due to Reynolds number similarities to the real world. These water tanks are highly repeatable and offer difficult measurements that can be even impossible to make in the field or by other means. So our experimental setup is shown on the right. You can see we have a laminar background flow of water pushed through the large flume, and we have a buoyant plume of dyed liquid released from the floor of the flume that drifts in the background flow, similar to how a pollutant would drift in the wind. And by varying the background flow, we can test the effects of different wind speeds on pollution dispersion and concentration. So using a stationary camera, we recorded 4 key videos of the flume experiment. 2 of these videos have no obstacles, just a flat plane, whereas the other 2 have a single building obstacle. And this allows us to test both rural and urban environments. So each of the 2 sets of videos also includes a 0.04 meters per second slower wind speed and a 0.15 meters per second higher wind speed. So you can see here the 2 videos. There's one without obstacles, and here's one with the 1 building obstacle. So to explore these videos and gain some usable quantitative data, we used a Python code of our own to crop them to ground level, as you can see here, and extract 20 frames from the middle that we believe to be representative of the entire portion of the video. So we used a heuristic method to scan through all the pixels and separate the reddish pixels from the rest of them. And then we used another brightness heuristic along with some thresholds that we experimented with to divide these reddish pixels into light, medium, and dark pixels, where the dark pixels represent a higher concentration of pollutant and the light pixels represent a lower concentration.
We use Python's pyplot package to analyze the data gathered using the method described on the previous slide. As you can see in figure 3, the slower flow rate seemed to have a higher concentration of dark pixels compared to the faster flow rate for every frame analyzed in our investigation. We also used a 2-sample t-test from the SciPy package to add additional support to these qualitative observations. Shown in table 1, the p-values obtained from the t-test for the pixel concentrations of all saturations were all under the standard alpha value of 0.01, showing that there is indeed a significant difference in the average pixel concentration values between the 2 videos. Also looking at the mean concentrations in table 1, we can see that the dark and medium pixel concentrations decrease with a higher flow rate while the light pixel concentration increases. Using that same method, we found significant differences for all mean concentrations across the 2 flow rates, this time with a single building obstruction. Figure 4 shows that there is indeed a significant difference in the dark pixel concentration between the 2 flume speeds, which matches the trend shown for the video data with no obstructions. Table 2 shows mostly the same trends as the no obstruction data. Dark and medium pixel concentrations decrease with a higher flow rate, though light pixel concentrations also appear to decrease with a higher flow rate. From the results discussed in the previous slides, we concluded that a higher flume speed spreads out the particles more and in turn lowers the concentration of dark pixels. This seems to support the claims from past literature that an inverse relationship exists between wind speed and pollution concentration. However, we can see that the methods used to find the dark, medium, and light pixel concentrations are inherently heuristic, and the flume itself is idealistic, so it doesn't take into account different variables seen in real-world observations. Due to these limitations, a trend cannot be effectively predicted from experimental data alone, so we also decided to analyze real-world wind speed and pollution data from several cities across the United States to further support our findings from this experiment.
So like Prabhas mentioned, we look at the air quality index and the wind speed data for 3 pairs of cities across the United States, so 6 cities in total. We gathered wind speed data from a site called Weather Underground and the AQI data from the US Environmental Protection Agency. So to clarify, AQI is a measure of how polluted the air is. It takes into account 4 major pollutants: ozone, particulate matter, carbon monoxide, and sulfur dioxide. Low AQI values are considered good, while higher AQI values suggest poor air quality and can pose health risks. So the 3 pairs of cities that we chose were San Francisco and Los Angeles, Boston and Philadelphia, and Tallahassee and Orlando. We chose these pairs because they were similar in climate, region, and urbanization. However, they also have very different wind speeds, and thus we predicted to see different AQI values to match the inverse relationship that we observed with our experimental data. We took our data from 2019 because we wanted to choose the most recent year whilst ensuring that the data wasn't affected by any COVID factors. To ensure the wind speed data and the AQI data were significantly different, we ran a series of t-tests between the data for each pair of cities using Python code.
The p-values returned for the t-tests we ran on the Los Angeles and San Francisco data are shown in Table 3, with both the p-value for wind speed and for the AQI being below 0.01, meaning that there exists a significant difference between the data for wind speed and AQI for Los Angeles and San Francisco. Figure 5 shows a scatter plot of wind speed measured in miles per hour on the x-axis and AQI on the y-axis, with each point being one of the 365 days of 2019 that we analyzed. The red points belong to Los Angeles and the blue points belong to San Francisco. As you can see, both cities showed a general inverse relationship. As wind speed increases, AQI seems to decrease. As you can see in Table 4, the t-tests we ran on wind speed and AQI of Boston and Philadelphia were also returned significant p-values, illustrating the statistically significant difference between both the wind speeds and AQI of Boston and Philadelphia. Similarly to in Figure 5, in Figure 6, we can see a general downward sloping of the graph, although nonlinear, where AQI decreases as wind speed increases. For the Tallahassee and Orlando data shown in Table 5, we discovered a significant p-value for the wind speeds, but not for the AQI. This suggests that while there is a significant difference in the wind speeds between the two cities, there is not a significant difference in the AQIs. In Figure 7, which plots AQI against wind speed, there's again a general inverse relationship between the two variables, where the data reflects a similar AQI between the two cities, but different wind speeds.
Through analysis and comparison of the collected data, we found that our initial hypothesis is partially supported. Firstly, the video water flume data showed higher concentrations of dark-colored pixels at slower flow rates and lower concentrations of dark-colored pixels at higher flow rates in both the no obstruction and the one obstruction simulations. This is the exact relationship we anticipated within the water flume experiment, as it backs the prior literature on pollution dispersion and wind speed. On the contrary, though a significant effect of wind on pollution concentration was present in the Los Angeles, San Francisco, and Boston, Philadelphia data, the AQI difference with the Tallahassee and Orlando pair was not significant, such that the results of our flume simulation did not perfectly extend into the real world. This is most likely due to the flume's idealistic nature. To better understand the specific relationship between the datasets, we would need to collect a larger pool of city pairs, their AQIs, and average wind speeds. Further limitations include the potential confounding variables created by the observation stations which collected the real-world data, which were, albeit mostly similar throughout. A standardized method of collecting this type of data would have given us more reassurance in our results, or would have potentially shown different ones. With more time, we also believe it would be important to consider the other variables that may affect pollution concentration, such as temperature and weather in more depth, and to look at similar correlations in more expansive residential zones, such as counties. Although the hypothesis is only partially supported, we do believe the data we have collected and sifted through still serves as a good warning sign as to the future that might be without science delving deeper into the topic. Here's some sources we used. And lastly, before we begin accepting questions, our group would like to acknowledge and give a special thanks to Dr. Hancock and the Geophysical Fluid Dynamics Institute for providing us the fascinating video data we used, Ms. Ashley Gannon, Ms. Jamaica Greenwood, and Ms. Darren Sago for their invaluable mentorship and help with shaping our research, and the FSU Young Scholars Program and its inspiring directors for giving us this opportunity. Thank you once again, and if anyone has any questions, please ask them.
Thank you so much, Leonard, and everyone else. This was a great presentation. Let's give our last group a big round of applause with the reactions, and I'm happy to field a few questions for them. Okay, it looks like we do have a question in the chat from Vishal. How do you think the p-value you collected will change depending on whether or not more cities and wind data were included?
Yeah, that's a great question, Vishal. So we think that if we were to include more cities with more reliable data, then hopefully we would see similar relationships as we saw in Boston, Philadelphia, and as in Los Angeles and San Francisco, rather than the insignificant relationship we saw in Tallahassee and Orlando. So hopefully with more cities to check, we'll see more significant relationships. But it's also possible that we could see a lack of significant relationships, showing that the flume data was indeed idealistic, and we need to consider further factors before extending it to the real world.
Thank you so much. We do have time for one more question if anyone has one last question. Okay, Dr. Heister.
So do you think the plume would forever have this limitation? So 2 questions. And are there some adjustments that you could make to the plume that might help? And also, do you think there would be space for computer simulations to help broach that divide between the plume and the real world?
Great. Thank you. That's a great question. So the first part about adjusting the plume and the flume, I think there are definitely ways to do that. So we were thinking about having winds or flow rates that map to the wind speeds we saw. So for instance, if we could see an equivalent ratio to the ratios of the wind speeds we saw, then we could also see how the ratio of the concentrations compares to the ratio of the AQIs, which means we could actually map one to the other in a more effective manner. Plus, our urban environment that was only a single building obstacle, it could definitely be beneficial to include more complex models of urban environments with more appropriate building sizes. Testing different things like that would be great. And as for computer simulation, from our research, we found that computer simulation is pretty tough for something like this. Even just normal wind flowing through buildings can be difficult. And once you factor in a buoyant plume that's moving, it gets really complicated. But we do hope that if we had more time, we could look into different methods such as like SimScale or different Python methods we saw, and maybe experiment with some of that and see if we could get any good results from those.
Awesome. Yeah, modeling chaotic systems is really, it gets difficult really quickly, even when they're pretty simple, but that would be an interesting way forward. Okay, with that, I think this ends our formal presentation. So please use the Zoom reactions to give the students one last round of applause for the hard work. Give yourselves a round of applause. And I will turn the floor over to co-director and captain Barbara Shoplock for closing remarks.
I'm very excited, but now I have some professional closing remarks outside of just my excitement. This year we have over 200 students apply for these 40 positions, and all of the applicants met the rigorous YSP baseline requirements, but the students that you just heard present are the ones that easily rose to the top of the list. I mean, y'all are really truly the best of Florida's best. Most of you ranking in the top 5% at your high school, and your collective unweighted GPA is 3.97 out of 4. So I have to just say, wow, just wow. And given these types of statistics, you basically think that this might be a really cutthroat bunch of young people getting together, because now you're in a room with all the best of the best, but that just wasn't the case this summer. So we're particularly proud of your positive attitudes and the collaborative spirit that you brought to this program, and also the tenacity and perseverance that you exhibited in the face of this summer's unique circumstance. It's hard to be on Zoom all day, all summer. So, and we're confident that you will go on to do really great things. The talks you heard today here are evidence of what you can achieve and how impressive you really are as students. So in addition to all of our Zoom reaction buttons, I want everyone to go ahead and unmute and give an out loud round of applause to these truly inspiring students. So let's do that right now. Well done, young scholar. Make some noise, people. That includes you, scholars. You can like clap for other people. Oh, this is awkward. It sounds like 2 people, but I know we really are. And I'm really, really glad that we recorded these presentations, because my inbox has dozens of requests for the links for these presentations. The middle of the day is a tricky time to be able to do something. We are trying to respect the confines of our program, but I'm glad that we can share the links with all the faculty members that have time conflicts during this. So your presentation will continue and people might have questions. Before we go, I would like to call out and specifically acknowledge the efforts of one more person, a very important person. A very huge thank you to Dr. Erica Staling for bringing your significant expertise to the program this year, both in terms of as a scientist and as an educator. Dr. Staling, Erica, I'm looking right at you. Your tireless efforts, creativity, and hard work were a statistically significant reason that this 39th iteration of the program was a success. Your ability to bring so many resources, to these lucky, deserving students during these 6 weeks is really remarkable and appreciated. Lots of work there. So students, I want to hear you. An out loud round of applause for your fearless research leader. So not just clapping, because that doesn't translate. So let me hear some woohoos. Claps just don't translate really well. So young scholars, the 40 of you, you are close to that finish line. You need to rest well tonight and study hard for tomorrow's finals and presentations. And we co-directors will see you at the final award ceremonies tomorrow at 2. But this officially concludes the 39th YSP Research Seminar. And thank you for all those who could come and celebrate the hard work. And for those of you who view this later at your own time, celebrating the hard work of these amazing students. And I genuinely mean that. Well done.
Thank you, everyone. This was great.